Hi everyone, I'm Raghav and I'm from University of Missouri, and I am interested in understanding RNA's role in the origins of life. And I'm basically concerned with how RNA catalyzes biologically relevant reactions. Thank you, Kristen, for the RNA World Hypothesis intro. That was really good. So basically, the RNA World Hypothesis says that during the early origins and evolution of life, it was the RNA that was the workhorse, acting both as a catalyst and also as a genetic information carrier, which is, of course, now done by the DNA and the protein. Although originally it was put forward to circumvent the chicken or egg paradox, whether DNA came first or the RNA came first, well, recently we've actually found many catalytic RNAs by in vitro evolution, and it has really strengthened this hypothesis. So I'm interested in phosphoryl transfer and how RNA catalyzes phosphoryl transfer. For example, well, in modern biology, what we have, okay, so in modern biology, we know that all the phosphorylation reactions are actually catalyzed by protein kinases. And in case of the RNA world, you'd think that there would be kinase ribozymes or RNA enzymes that can catalyze this reaction. So how do you find catalytic RNAs that have this activity to transfer phosphoryl group? So we are actually selecting for RNA enzymes that can phosphorylate themselves. So they take the phosphates off of a phosphoryl donor and then transfer onto themselves. So that's what they would look like. And the way you can find these active RNAs, the way you can partition the active RNAs from the inactive RNAs is, so if you notice carefully there, the phosphoryl donor actually has a sulfur instead of an oxygen on the gamma position of the triphosphate. So any RNA that is active actually gets a sulfur along with the phosphate. So if you separate these RNAs out on a polyacrylamide gel where one of the layer in the middle contains mercury, the sulfur interacts with the mercury, and then all the RNAs that have the sulfur get trapped in the mercury layer. So if the RNA is active and if it has the activity of a kinase ribozyme, then it gets the sulfur along with the phosphate and gets trapped in the mercury layer. So you can basically cut out this band that is trapped in the mercury layer, reverse transcribe it, and then PCR amplify it, do this process over and over again until you finally enrich your pool with active RNA. So essentially, you start out with 10 to the 14th different sequences, and you end up with a fraction of them that have the desired activity. So this brings to part one of my talk, which has to do with understanding how this ribozyme catalyzes phosphoryl transfer, what kind of products can ribozymes form off of this reaction. So this ribozyme was actually selected in my lab. So the GTP gamma S is the phosphate donor. It uses magnesium and copper and phosphorylates at this second guanosine position. And so this is basically what a polyacrylamide gel would look like. As the time increases, you see more and more RNA getting phosphorylated and gets trapped at the mercury layer. Okay, so one of the first things I wanted to understand was how does the 2' hydroxyl in an RNA, in this particular ribozyme, play, what kind of role the 2' hydroxyl plays in this RNA structure. So we know that 2' hydroxyl is involved in different things, like a lot of different things. For example, here it's shown coordinating a metal ion, and it is also involved in hydrogen bond as a hydrogen bond donor or a hydrogen bond acceptor. So it is very important to form the RNA structures, the active RNA structures, and ultimately give RNA the catalytic activity. So to figure out whether this 2' hydroxyls in this RNA is important, what kind of 2' OHs are important in this ribozyme, what we did was we tried to substitute 2' we try to substitute the nucleotides with 2' fluoronucleotides. So you basically transcribe this ribozyme with either ribonucleotides that have 2' hydroxyl or ribonucleotides that have 2' fluorine. So we either made the RNA with all 2' hydroxyl or substituted all G's with 2' fluoro or all A's with 2' fluoro, all C's or all U's one at a time. So again, the same idea, 2' hydroxyl, the RNA without the phosphate donor, the RNA, of course, doesn't get phosphorylated. Once you add the phosphoryl donor, it gets phosphorylated, so it gets stuck at the mercury layer. And we were really surprised that when you replace all these ribonucleotides with 2' fluoros, they still get phosphorylated and the ribozyme is still active. So we wanted to know more about this, this ribozyme, because what this tells us is that actually no 2' hydroxyl is important for this, or no particular 2' hydroxyl is important for the activity, because I also tried making the ribozyme as a complete deoxyribozyme, and that is dead. So of course, like if you start replacing multiple 2' hydroxyls from multiple nucleotides, you lose the activity. So initially what we had found was that for the ribozyme, that with the 2' hydroxyl, the rate of the reaction increases as you increase the pH. And one obvious explanation to this would be as you increase the pH, the 2' hydroxyl gets deprotonated, and that somehow increases the rate of the reaction. So now that I had all the 2' fluororibozymes, I tested all these ribozymes with the 2' fluorine positions at different pHs as well, and they all showed similar behavior, and they all were being stimulated at higher pH as well. Now, what this tells us is that it's not the 2' hydroxyl that is being deprotonated, but there is something else going on. And we were really intrigued by this data because all this time we also thought that it was the 2' hydroxyl that was getting phosphorylated, but it might not be the case anymore. So to answer that question, what we started to do was, so this is the full ribozyme. What I did was I basically designed a transacting version of the ribozyme where the part that gets phosphorylated is actually separated from the catalytic strand. So you bring these two molecules together, the catalytic strand base pairs with the substrate strand and phosphorylates at this guanosine position. Now what this design enabled us to do was we could actually test different substrates now. And that's basically what I started out doing. First of all, I used an all RNA backbone as the substrate and all DNA as the backbone as the substrate, and then a DNA RNA hybrid substrate as well. And to our surprise, everything, all of these three substrates got phosphorylated. Now, what is really intriguing was that for the second guanosine, the only obvious phosphorylation site is the 2' hydroxyl. And in a DNA, we know from biochem or biology 101 that there is absolutely no 2' hydroxyl. So this is actually first observation of direct nucleobase phosphorylation by a kinase ribozyme. Now, why this is important is because once you start modifying nucleotides, an early prebiotic Earth would have more nucleotides to sample from. It would have, that's basically a way of generating different kinds of nucleotides so that nature can sample to see which one is optimum for life to use. So if you actually go back to the structure of this complex, everything from the substrate is actually base paired with the ribozyme. So that's actually pretty boring. It's just a DNA RNA helix. There's not much going on. But if you look at the business end here, this GGA is this 5' GG and A. It's free, and it is free to do stuff. It can actually interact with other parts of the ribozyme. So I started out testing whether you can change this GG and A, so that's GGA, to other nucleotides or not. So if you mutated this G to a C or a T or an A, the ribozyme is dead. So these are very soft mutations where you're only changing some atoms. So in this case, you're going from G to an inosine, where you're only taking this amine out. And for this G1 and G2, pretty much any soft mutation that you do, the ribozyme is dead. Like it absolutely is very specific to this GGA sequence. Now this will be very important. We'll come back to this again for the second part of my talk. We basically then wanted to understand what kind of product is this ribozyme forming. So to answer that, I basically took the substrate, did not react with the ribozyme, but added a phosphate on the very 5' end of the ribose sugar. Took this substrate, incubated it at different pHs from 9 to 4.5. And you can see that the product that has the phosphate is still stable at all this pH range. Next, what I did was I took the same substrate, now this time added the phosphate using the ribozyme. And we know that the phosphate is on a nucleobase. It's not on the 2' hydroxyl, but we don't know what atom the phosphate is attached to. Now you take this product formed by the ribozyme and do the same experiment, incubate it at different pHs, the phosphate actually starts to come off. So chemically speaking, the ribozyme forms a different product than what you'd have if it was just a ribose phosphorylation. And then I started digging out literature to see like what people have done. This is like, I don't even know when this paper was published. Okay, it was from 2004. But what this paper essentially said was that in acidic conditions, the PN bond, they break faster than PO bond. So what this suggests is that the phosphate might actually be linked in one of these two atoms. So we still don't know where the phosphate is linked, but it is pretty interesting that we're starting to see this novel targets for RNA-catalyzed phosphorylation. So that's the sort of summary of the first part of my talk, I guess, is that this ribozyme phosphorylates the nucleobase directly, and the phosphoryl acceptor is likely an N. We don't know whether it's an N2 or an N1. And obviously, this is the first evidence of direct modification of nucleobase by an RNA enzyme. And as I said earlier, nucleobase modification would have been a nice way to explore the chemical space for nucleotides in an early RNA work. So this brings to sort of the second part of my talk. And so you have all these ribozymes that people select, and they say, oh, we found this ribozyme, but they just ended at that. What are you going to do with it? So I was looking for ideas to use this ribozyme to regulate other RNAs that are of biological or astrobiological significance, I guess. So if you look at the ribozyme structure with a substrate, everything else, as I pointed out earlier, is base paired with a substrate, which is pretty boring. Like in principle, you should be able to change all these base pairs, and the ribozyme should still be active as long as this part is kept constant. And so that's basically what I was studying. So one of the functional RNAs that I wanted to target using this ribozyme was the ATP aptamer. So this RNA molecule actually binds to ATP, and it was discovered by Jack Szostak in I think it was early 90s. But if you look at the sequence of the ATP aptamer already has a GGA, and that's why I wanted you guys to keep in mind about that 5' GGA terminal, because the ribozyme absolutely needs it. And turns out that the second G that the ribozyme would modify it actually makes direct contact with the ligand AMP, acting as a hydrogen bond donor. So we wanted to see whether we could design a ribozyme that would phosphorylate ATP aptamer, and what would happen to the ATP apter function. So this is, I guess the diagram kind of messed. I'm sorry about that, but you can't basically say anything. But anyway, we designed the ribozyme, and turns out that you can phosphorylate the ATP aptamer. And as you can see, the phosphorylated ATP aptamer, again, is retained in that mercury layer. So the next thing I wanted to study was what happens when this ATP aptamer is phosphorylated. Is it still functional? And the way you can test that is you have a column with agarose that has ATP embedded in it. And what you do is you add ATP aptamer, the RNAs that bind to ATP bind to the column, and the unbound RNAs basically flow through, and you wash multiple times, so all the unbound RNAs is eluted out, and then you add free ATP molecules so that the RNAs that are bound to the agarose now grab onto the free ATP molecules, and then they elute out. So this is basically what I did. ATP aptamer without the phosphate, you add to the column, then you start washing, they come off, and then, they come off, and then there's a certain amount of ATP aptamer that is still bound to the column. And then when you wash it with ATP-containing buffer, then the aptamers bind to the free-floating ATP, and then they elute out as well. The story is different when the ATP aptamer is phosphorylated. The aptamer can no longer bind to the column. So this could be a very interesting way of regulating functional RNAs in an RNA world, because if there is a functional RNA, if there was a functional RNA molecule that bound to NTPs, and if there were ribozymes that started phosphorylating these functional domains of these aptamers, then the ribozymes or the aptamers would no longer be functional. So this brings to, so then I started looking at whether, okay, you can phosphorylate ATP aptamers. Can you phosphorylate longer RNA chains? And this is sort of like a background of why we wanted to do that. So this is just a simple primer extension assay that I'm showing here. You have a template, you have a primer, the primer binds to the template, and then you can add a DNA polymerase to extend it. Whereas if you have a phosphorylated template, you add the primer, anneal it, extend it, but the extension stops right where the phosphate is. And this is basically what this just shows. Primer, extended primer. Once you add the phosphate, it stops right where the phosphate is. You can make the template longer. Again, you see extension, but as soon as you add the phosphate, it stops right there as well. Now, what I was trying to get at was you have mRNA sequences that might also have GGA in them. So what happens if you phosphorylated a G, and while the ribosome is trying to scan through this mRNA sequence, what does it do when it encounters modified nucleotide? And we know that the codon-anticodon interactions are primarily governed by the Watson and Crick base pairing, and when you're phosphorylating nucleobase in G, you're essentially messing up with that Watson and Crick base pairing, and I think this might be a really good way to turn off genes as well. So to answer that question, we started generating new ribozymes that would target sequences of a longer RNA chain. So this is an enhanced green fluorescent protein mRNA sequence, and it turns out that it has 11 sites that have GGA sequence in them, and I think everything has shifted, so you can't really see. There's one, but there are 11 sites that have GGA in them. So we basically generated ribozymes to target each of these 11 sites, and turns out we can actually phosphorylate multiple sites in enhanced green fluorescent protein. So what we're going to do next is we're going to take this phosphorylated mRNA and do an in vitro translation experiment to see whether this phosphorylated mRNAs are translatable or not. If they are, well, what we hope is that the translation is shut off after phosphorylation, and this would mean that phosphorylation by ribozymes could be a new avenue for artificial genetic regulation, and possibly could be used in synthetic biology as well. We're also working on to reverse the phosphorylation. The first part of my talk, I showed that at low pH, you start losing the phosphate. So we're also trying to explore what happens once the phosphate is lost. Can you regain the functional activity that you initially had as well? So with that, well, this is a summary of the second part. Well, functional RNAs can be targeted for ribozyme-mediated regulation of other functional RNA molecules. So you're basically, so this is literally making like an RNA world, because we're controlling activity of one functional RNA molecules with another ribozyme. And mRNA molecules, mRNA phosphorylation may potentially be useful for artificially regulating gene expression. And this basically means that metabolic networks in an RNA world could be mediated by ribozymes. And with that, I think that's it. Yeah, I'd like to thank my lab and, of course, you all for your time as well. Thanks.
Any questions?
Thanks for that. It was an interesting talk. Now, you mentioned a little bit before the relationship between like proteins and sort of RNA enzymes. And when I think of a protein, the active site usually has a metal there, right?
Right.
So how does this work with RNA enzymes? I mean, do they incorporate a metal at some point?
Yes. Well, some RNA enzymes, they utilize, they require metals, absolutely. But there have been some that are metal independent as well. This one particular actually uses magnesium and copper for some reason, but yeah.
Just a quick follow-up. Do you know how those metals are incorporated? I mean, are they kind of dynamic? Like, do they come in and go out, or are they fixed? Like when you have this kind of structure, that it's almost like a protein.
It's pretty much fixed, just like proteins. There's specific metal binding sites, metal ion binding sites, and these metal ions usually interact with the 2' hydroxyls as well, and so it's really important for the RNA folding.
Thanks.
Yeah.
A quick question about your first part of your talk.
Sure.
So you were showing the function of your ribozyme against pH.
Right.
And I noticed the range that you did was a little bit tight. Mm-hmm. Did you try pushing it very far, out of curiosity?
Actually, yes. So that work had already been done before, and I was basically only testing it at a small range. And yes, it is evident in a larger range as well. I think we have gone from 5 to 8.5 or something like that.
Thank you.
Thanks.
Other questions? Maybe some from SegaNet? No? Anybody else? No? Then with that, thank you, Raghav.
All right, thanks.